Hello. Let's talk about HIV entry. Make sure we're talking into the microphone. All right. These are all the parts of the HIV virion, our little mascot for today. This is the glycoprotein of HIV. So this is the envelope protein, the thing that sticks to your cell. The little purple dancing things are all the sugar molecules on there. It's a glycoprotein. Those are glycans. They're just different kinds of sugar molecule chains. And these are common on proteins you would find outside the cell. And they have the effect in HIV of being a sort of camouflage because they keep, changing around, probably something like that. And it results in the immune system not ever being able to get a good look or a good lock on HIV. So it kind of goes undetected. It's very difficult to make a useful immune response. I believe the red parts are all the parts that are going to bind to receptors. But let's talk about that process because there's a lot of stuff to unpack here. All right, so we're talking about this protein, and what you can see over here is mostly the top of the protein. The transmembrane region, which is the bottom of this thing called TM, is generally not going to be solved in the same crystal structure as the top, just because hydrophobic domains are ridiculous, honestly, to solve. They have their own methods. They're a big problem. All right, so here's our HIV virion. You can see some of these glycoproteins, these envelope proteins, all around the top. And then inside, you're going to have that little capsid, which is going to be made of the CA protein, capsid protein. And then inside the CA protein, there's going to be a nucleocapsid protein, wherever that is. You're also going to have a couple of these other proteins, the reverse transcriptase, the integrase, and the protease floating around in there. The matrix protein is going to be lining the underside of this membrane. So it joins on to the tails of the glycoproteins, the envelope proteins, as they are called. Yeah. Oh, here's our nucleocapsid. There we go. And yeah, it's on the inside. All right. So, the story of HIV entry has several steps. The first step is that the virion, which is the little spiky ball, with the envelope proteins coming out of it, is going to bind to a cell. And it binds to a receptor that's called an attachment receptor, which means HIV can stick to it, but it is not sufficient for HIV to actually enter the cell. For that, you need an entry receptor, or you can call it a fusion receptor. And it's going to end up being a totally different protein, but one which is going to be common on the same kind of cell that has the attachment proteins. So it works out fine for HIV if you were worried. So here we are, we're binding to the attachment protein. That is going to be binding to a part of the protein that sticks up out of the cell. And once it is bound to that, then it has more time to sort of bounce around on the surface of that cell and worm its way down and eventually meet the entry receptor. Because while you see an image here that's showing all these proteins just, naked and alone on the surface of the cell, it's not like that. There's an extracellular matrix, which is strings of protein and sugar strands, and it's kind of like a little coat that the cell is wearing all the time. And so it's only the real little tiny tips of some of these proteins that'll actually poke up through that coat. And so that is the part that HIV is going to make first contact with, for sure. So, despite what you may have learned in, high school, maybe even college biology, HIV-1 is going to attach to proteins like CD4. Also DC sign, which is found on dendritic cells, and also L sign. It can attach a little bit to heparan sulfate, which is a kind of sugar that is found on some proteins. And it can theoretically attach to a thing called a glycosphingolipid. So this is a lipid molecule, like part of the membrane, but it has a sugar molecule attached to it. Weird little things. The thing with that is that those, I believe, are fairly short molecules, so they're not going to poke up through the top of the extracellular matrix in most cases, unless you get a weird cell with a bald patch, which, hey, happens to the best of us, right? Yeah. Yeah, that's what the virus is trying to do. It attaches to any of these attachment receptors, but most commonly CD4. That doesn't get it into the cell, but then it will bind to one of these two receptors. And there are a couple other things that it can bind to a little bit to get in, but not as well. It seems to be CCR5 and CXCR4. CCR5 would be common on macrophages, which are very common in the bloodstream and are the main cell type that HIV is going to bind. CXCR4, I believe, is going to be found on CD4 helper T cells. So that's how it gets into both of those. All right, let's carry on. And those are both involved in immune signaling. So the next step is to make a little map, at least in your mind, of what this envelope protein is. So this thing, dancing around up there. It's got a lot of parts, and some of these parts are very important in the story. They're all important at different stages, but I'll call out the ones that are most important. So at the start, you have a thing called a signal peptidase cleavage site. So that's a little protease, like thing that cuts protein, and it's going to be found in the cells, and it works exactly like ancient movie tickets or train tickets used to work. You'd go in, you'd show them to the conductor, I guess, and they'd tear them in half, and you keep your half, and that would get you permission to go into the next, the next car on the journey, whatever. With the signal peptide, it only has to be about 14 amino acids long, the small ones. And it's a little hydrophobic transmembrane region. It's going to go through, and then it's got a couple of little small residues that are a little bit polar, and that's where the cut is going to happen. And once that's cut, basically our protein has its ticket punched, and it will get transported. It'll get to associate with proteins that will transport it up to the plasma membrane, which means getting out of the ER, going through the Golgi apparatus, which sounds like a lot of fun, in the cis side, going through the medial, out the trans side, and all the way up to the cell surface. So that is what happens. And I don't know if you can assign reasons for these things, but the reason HIV is doing this is because there are proteins that it needs to add sugar molecules and trim back the sugar molecules on the surface and make it look just like our little dancing purple blob up here. And those are all those enzymes that are going to add and trim the glycans, the sugar molecules. Those are going to be found in the ER and in the Golgi. Most of the addition happens in the ER, where we grow these great big bushy glycans, and then we trim them down and cap them off with a thing called sialic acid once it's in the Golgi. That's not important for HIV, but it's going to be real important for flu when we get there, because that sialic acid, that thing that's added to every glycoprotein pretty much, that's going to be the receptor for the flu virus, which means, it can get into pretty much any cell with a glycoprotein, which is pretty much any cell. Yeah, so bad news. But that's in the future. Don't worry about that now. Let's focus on HIV. It's bad enough news. All right. The rest of the GP120, which is the first half of this, that's going to be the part that actually binds to the receptor. It's going to be the thing you see in yellow with the little red parts up here. That is going to be divided into, they call them constant regions or conserved regions. Those are the C parts, and it's got variable regions. Those are the V parts. Yeah. Did you figure that out? Yeah. It's not too tough. Each of these little stick figures on here is showing where one of the glycans is going to be attached. And so that's where we get all these little sugar molecules. The protein is going to form a trimer, so there's going to be 3 copies that come together and make this little structure. That's 3 of them, which is why it looks real symmetrical as it rotates around. Let's see. The important part is going to be the V3 loop right there, because the V3 loop is going to be the part that actually is going to bind to the entry receptor or the fusion receptor, which is going to be CCR5 or CXCR4. Yeah, that's just the name of it. Yeah, it's horrible. It's an acronym, but you can't do anything about it. That's just what it's called. All right. Yeah, so the V3 loop is our important one. Then we have a thing called a furin cleavage site here. Now, furin cleavage is going to happen outside of the cell where the virus was made. And it can happen either on the surface of that cell, or it can happen on when it bumps into a cell in transit to the next cell it's going to infect, or it could be cut by furin when it gets to the cell that it's going to infect, potentially. Yeah. And any of those will work, and to some degree, each of those things happens. So there's not like a defined point in the life cycle where you can say, furin has to cut right now. Yeah. That's going to be important because it's going to free up this little thing that's right here, which is called the fusion peptide. Fusion peptide is a little hook. That's like, like that Mortal Kombat guy. Just a little hook that's going to hook into the cell and crash the virus into the cell. The crashing is going to be done by these two things, heptad repeat 1 and 2, and we'll cover those in another lecture about fusion. And then we're going to have the MPER, that's going to bring some lipids from the virus membrane and pull them toward or into the cell membrane. And that's going to be the most important thing for hemifusion, which is like halfway fusion. It's that midpoint of fusion, which is actually kind of at the end of the fusion process. Yeah. And then the cytoplasmic tail does other things, but they're not things that we are going to have to worry about too much. The one big thing is that there is a spot on the cytoplasmic tail that is going to have a lipid molecule attached to the protein. So you think of proteins and lipids as being different things, but you can actually end up with sugars attached to proteins, lipids attached to proteins, all kinds of weird stuff goes on inside the cell. So this is going to be, we call it acylated, which means it has a lipid tail, an acyl group attached to it. And that's the case. Or you can say amino acylated because it's attached to an amino acid. Yeah, amazing. All right. I think that's enough of HIV. Envelope map. Okay. So there's a bunch of words here. You can read through those on your own. They're going to explain some of the stuff that we've got, and I pulled them out of this paper right here. And they're like the description for what we're looking at here, which that is this. It's the same thing, only they haven't drawn the glycans over here, and they have drawn the glycans and made them dance over here. Yeah, but pretty much the same. They're even both yellow. Yeah, if that makes you happy. So the first protein that's going to encounter HIV most of the time is going to be CD4. And remember, this is a marker for both macrophages and helper T cells and a bunch of other cell types in the body. Yeah. But all things that are connected with immune function, let's say. And so CD4 has 4 of these little domains, and you can see they're kind of the same shape. And we call that shape an immunoglobulin-like domain, because immunoglobulin is the fancy word for antibodies, and the antibodies are made out of chains of immunoglobulin-like domains. Doesn't mean this is an antibody. It just means like a couple million years ago, these two things were related. They stole from the same place. Yeah. So these immunoglobulin-like domains, there are 4 of them in CD4, and then there is a transmembrane region down here that they can't solve in the same crystal structure. It's the same problem you've got here. You can solve the top of it, but the part that's stuck in a membrane is just obnoxious to solve. Although I believe there's a decent cryo-EM structure for at least HIV envelope. All right, so CD4. The important part of this is going to be the tippy top, the bit that sticks out. And so phenylalanine 43 is going to be right around here where I'm dragging my cursor, and it's part of domain number 1. So phenylalanine, that's one of our 20 amino acids, 20 common amino acids that make up protein. So it's just the name of which part of the protein is actually doing the binding. If you remember your amino acids, phenylalanine is going to be one of these that's sort of like a hexagon, and it's got those distributed electrons inside. It's aromatic, is what we say. And that is going to make it super duper hydrophobic, and that is going to be sticking up. So you have this thing that's sticking up in water, and it's a very hydrophobic amino acid side chain as they go. So happy is completely the wrong word, but I tend to think of it that way. This is an unhappy amino acid. It's in a place where it would like to be buried in just fat, in like a membrane, something like that. It would be, yeah, just feeling great if it was there. And yet this thing is floating out in the water. And so what does HIV do? It comes along and it solves the problem. HIV envelope protein, GP120, is going to bind to phenylalanine 43 of CD4, and it's located on immunoglobulin, like domain 1 of CD4. There we go. And so this part of the envelope protein is called the cavity. You can say it's the phenylalanine 43 binding cavity. That would be a smarter way than what's written here, and I'm going to have to go and change that. And that is going to be our interaction. Yeah. So here you can see GP120, and the star is the little spot where phenylalanine 43 is going to fit into GP120. And so that's going to be somewhere over here on the part that sticks out, if I'm interpreting that correctly. And then the part up here, I believe, is going to be the V3 loop, and that's what's going to pop out later. So that is a hydrophobic pocket. And it's got Y is a pretty hydrophobic amino acid, I is pretty hydrophobic, V is pretty hydrophobic, M is very hydrophobic. And so those are going to make enough of a hydrophobic environment that it's actually energetically better, lower free energy, for that phenylalanine to be bound to HIV than it is to be floating in the water, and that's why it happens. No energy is expended in doing this. It's just that by bringing those two things together, they become stable because the hydrophobic part is not surrounded by water that's being repelled. Basically, that repulsion goes away. Yeah. All right. There are several small molecules that will bind to that same pocket on GP120. And so these can be used theoretically as antivirals. You can put something in there that blocks the little part of CD4 that would be binding to the HIV-1 glycoprotein, the envelope protein. All right, so phenylalanine 43 binds to GP120. Phenylalanine 43 is on CD4. GP120 is the part that sticks out of the virion. It's the top of the envelope glycoprotein. All right, structures of some other things. You've already seen this one. We don't even need that one. But here is what CCR5 looks like. This is the entry receptor or fusion receptor. And here is CXCR4. There's two copies of it. But you can see that little blob down there is this little blob down here, and this little thicket of alpha helices. This is the part that's going to go through the membrane, and that is going to be, these are going to be the entry receptors. You can see they're very similar proteins. These are both a kind of protein called a G protein coupled receptor. And those are the ones that we make all the drugs to. Those are the ones that bind neurotransmitters and all kinds of cool stuff. So they're called GPCRs, and they're neat. In this case, these are G protein coupled receptors that are involved in activating macrophages or activating those helper T cells. When they receive the correct protein docking into them, which is like a little messenger sent by another immune cell, then they'll get turned on and they will start to go out and attack whatever the right thing to attack is. So here is this bottom protein. The green thing is the same as this protein up here, but it's bound to its ligand, which is called CCL5. That's the protein that CCR5 would like to bind. Yeah, so that's the story. And then the things that are a little harder to see here are that you're going to have movement. Whenever the ligand binds to these G protein coupled receptors, there's a lot of movement in its alpha helix number 5 moves a lot, and you get a little movement in alpha helix number 7 and 8. Those kind of swing and move. and so that kind of latches. It's like a Venus flytrap. It sort of closes down on the thing, and so that's how once it finds the right molecule to bind to, it stays stuck to that. And these are going to be, of course, signaling proteins. They're going to set off a big response inside the cell, and that's then going to change what the cell is doing. And in this case, it's going to drive the cell to start to reproduce itself and become activated and, kind of ramp up to go fight some kind of infection. Yeah. Which is unfortunately what HIV wants here. Yeah, that's the dirty part of it, because HIV gains more out of that process than it loses. We'll get to that later, but it's a rotten virus. So here you can see these are all CCR5 here, and here is CCL5. So this is the protein that's supposed to bind to CCR5. It's the little message from the immune system, and it's going to bind in there. And there's a little alpha helix, little twisty alpha helix that I'm tracing right here, and that's going to reach out, and you can see a couple of these little side chains. They just put them in here on this helix, but they're going to make looks like 4 points of contact inside this little pocket. So this pocket is in between the transmembrane alpha helices of CCR5, which you can see here. So that's going to bind. It's going to cause that sort of closing of CCR5. It's going to lock down onto it. That's the normal situation. HIV is going to use this, of course. Yeah. So HIV invents nothing. It just steals things that are lying around and uses dirty tricks to impersonate other cellular proteins in this case. So when GP120 binds to CD4, that whole thing that we talked about before, that is going to loosen up this V3 loop, which is going to stick out pretty much on top of the protein. I believe it is this red part that's right here. That V3 loop, look at that, has a little alpha helix right there. Now, the alpha helix is at the end of this protein, and it's kind of in the middle of this protein. But darned if it doesn't make 4 little points of contact inside the pocket, just like CCL5. So HIV is mimicking this cellular protein, and it's doing that because that will make CCR5 close down on HIV now, and HIV will be latched onto the surface of a macrophage or a helper T cell. And once that happens, then it's bad news, because that cell is probably going to become infected, though there are still a couple more chances for us to just barely escape infection, but usually not. All right, so here is a structure of, this is 1, 2, 3, 4. Those are the domains of CD4. You can see they're bound here. Here is this blue thing that's sticking down inside. It's kind of behind everything. That is the V3 loop that's sticking down. And this blobby red thing is CCR5. Here's a nice little interpretation of what's going on here. And so that is exactly how it looks, with your binding pocket being over here and your V3 loop being on the top. They're just on different faces of the same part of GP120 envelope protein. All right, and binding is going to change the structure of GP120, and it's also going to change the structure of CCR5. CCR5 closes, GP120 kind of loosens after the phenylalanine 43 of CD4 binds, and that loosening is going to let the V3 loop come out, and then once the V3 loop is out, then it's going to get attached very tightly. There are some other conformational changes that are basically going to let GP120 move out of the way, because once it has locked onto this receptor, GP120 has finished its job. It's done a great job. Congratulations, GP120. You are responsible for another HIV infection, basically. Okay, the last little part we'll do is what happens after that. So normally, when a cellular receptor like CCR5 gets a protein bound to it, it's going to latch down on it, just like we said, and that's going to signal for the cell to then pull this in. And so the cell is going to make a little bulge of membrane. It's going to pull it inward into the cytoplasm. And it has a little protein that does this. So it's the clathrin system. Clathrin is going to assemble this little cage, and it's this neat little three-legged structure. That's just what it looks like. And it assembles a little cage, like a little tiny soccer ball, around that part of the membrane. So as the membrane curves and gets pulled in, then clathrin binds some more. And once that is bound, then there's another protein called dynamin that's going to act like a little whip. It's going to wrap around and constrict that opening, and it's going to break this membrane here so that the plasma membrane stays on the surface, and our little, what we call a vesicle, like a little blob of surface membrane, is going to start its journey in toward the heart of the cell, if you like. Cells don't have hearts, but they do have lysosomes, and so that's where we're going. Yeah. And so this process, we're going to make a clathrin-coated pit, and then once the clathrin-coated vesicle has formed and dynamin has cut it off, that's the signal for other proteins to help the clathrin disassemble. And so now you just have this little blob, this little bubble that has an HIV inside of it. And that's going to be merged with some other vesicles, and there are whole intracellular pathways that do that, let me tell you. And that's going to then start the next part of our process, which is going to be the acidification part. And that's a good point for us to break, because this video has been long enough already. And yeah, I can't talk that long. I don't know how you can listen this long. At least you've got a pause button. So thanks very much. That's the end of this part of entry, but by no means the end of entry. There's a lot more entry to go. All right, stay tuned.